Hello, Genetics 311 students. Today we will be discussing point mutations and their overall effect. As we know, a mutation is a change in the DNA sequence. A point mutation is when a single nucleotide in the DNA sequence is altered. There are 4 types of point mutations. To show the effects of point mutations, we will be using this DNA sequence as our reference. Please pause the video and become familiar with it. Also note we are transcribing the coding strand of DNA. Therefore, the mRNA will be the same except the T's will be used instead. The first type of point mutation is missense. A missense mutation is when a single nucleotide alteration results in a codon that codes for a different amino acid. Referencing our normal DNA strand, we can see the base C change to an A. This is a transversion because the base change is from a pyrimidine to a purine. The effect of the missense mutation is the codon encodes for the amino acid aspartic acid instead of alanine. The second type is nonsense. A nonsense mutation is when a single nucleotide alteration results in a codon that encodes for a stop codon. Therefore, the translation of the sequence will end earlier than normal. Again, we have a transversion because the base C changed to an A. The effect of the nonsense mutation is the codon encodes for a stop codon instead of coding for the amino acid cysteine. We can see the sequence translation is terminated early and there is no cysteine. The third type is silent. A silent mutation is when a single nucleotide alteration results in a codon that encodes for the same amino acid. The base C is changed to T. This is a transition because the base change is from pyrimidine to pyrimidine. The effect of the silent mutation is the codon still encodes for the same amino acid, asparagine. The fourth and final type is readthrough. A readthrough mutation is a single nucleotide alteration in a stop codon, resulting in a continuation of the sequence. The base A is changed to G. This is another transition because the base change is from purine to purine. The effect of the read-through mutation is the stop codon is now a codon encoding for an amino acid. We can see the translation of the sequence continues, and the codon now encodes for the amino acid tryptophan. Now let's put all the pieces together and analyze the overall effect of point mutations. On the DNA/mRNA level is the point mutation, where one nucleotide is changed. On the amino acid level, the effects we talked about. And now the protein level. A protein's function may increase, decrease, not change, or not function at all. For example, read-through mutations cause the polypeptide chain of amino acids to be extremely long. This often results in a non-functional protein. For another example, silent mutations often go unnoticed because the same amino acid is coded for. Therefore, there is usually no change in protein function. Thank you all for watching.